Hello. Now I will be audible.
Yes, ma'am, you're
Yes, ma'am. Now
audible.
you are.
Okay, okay. So without further ado, we can start now. So a very good evening, everyone. I hope you all had dinner. So we will start from this unit. That is introduction to the human microbiome. So basically in this unit, we will study the exact composition of the various microorganisms residing in our body, right from top to bottom. Wait, let me connect my notepad. Okay. So in this unit, you will study the overview of the gut microbiome, composition and function. So before directly jumping onto the gut, we will see the overall organ of the human body, like from mouth to the intestine, large intestine, small intestine, and the different type of microbes residing in our body and the different kind of ecosystem, like in our different organ different pH is there, in the intestine different pH is there, different environment is there. So what kind of pH triggers what kind of bacteria or microorganism. So we will study, and majorly we will study the factors influencing gut microbiota, that is diet, lifestyle and environment. So let's start. So we will study this in every possible detail so that you will be able to call what is microbiome, what is microbiota, and what are the different microbial diversity in our body. So let's start. Okay. So microbiota. What is microbiota? So as you all know, our body have billions and billions of bacteria. Not only just bacteria, but also some different organism which are synergistically living in our body. It means a human body, you can see in this figure, every part is colonizes by different kind of microorganism. Now, can you just tell me that whether it is harmful or beneficial? Most of the
Beneficial.
beneficial. Most of the time it is beneficial. When it will be harmful? When the environmental factor will transform the normal microbiota or normal microbiome into something other that anyways hinder our normal metabolic machinery of any individual. So microbes that coexist. In biology, we always have to focus on the terms, like I have written here coexist. Not why not only exist? Because they are coexisting with the normal metabolic machinery of that particular individual. Suppose in my body, I will be having normal microflora. So normal microflora means microorganisms are residing in my body, in an human body. So both these organisms are coexisting. Why they are coexisting? They are mutually existing in an environment to support the life. These microorganisms are sustaining life and they are easing the life for the human body, like to perform the daily metabolic machinery. They are also providing some immunological memory to the body so that whenever we encounter an antigen, it will trigger the immunity towards it. So सबसे the most major application you can say, or the major advantage you can say of the microorganism is that it provides health benefit. So in what way it provides health benefit? It provide health benefit through the immunology. So many different kind of microorganism triggers different part of the immunological system and thereby preventing the harmful infections. So what you can say, microbiota are the range of microorganisms that may be common cell, mutualistic, or pathogenic. So I have used three terms: common cells, mutualistic, and also I have used the term pathogenic. So this pathogenic is conditional. This is not applicable for all the microorganisms. This is conditional. Whenever some environmental factor will transform the normal microflora, then in that condition it will transform into some pathogenic strain. But most of the time, 99% of the time, it is common cell. Common cell means it will benefit the organism, but it will not harm the organism. Either it will benefit the organism or it will not cause any benefit, any harm to the organism, plus zero relationship. Common cell. Mutualistic means it will show a positive to the host. So most of the time the microbiota, you can just remember these three terms, are the range of microorganisms that are common cell, mutualistic, and pathogenic, which are found in a multicellular organism like human body. These are the part of normal flora. Flora means not plant. Here it refers to the different kind of microorganism residing in our body. So they are of different types. Some are anaerobes, some are aerobic, and also their condition they required to grow is different. Somebody's require alkaline pH, somebody require acidic pH. So accordingly they reside different organs of our body. And the majorly why we are focusing on the gut, because the major diversity or the major abundance you can see is in the gastrointestinal tract. Therefore in the microbiota we always focus on the gut. You have heard about the term, if gut is okay, your whole health is okay. So this is why we are focusing on the microbiota or the gut microbiota. So you can see variety of microorganisms are living in our body. They are coexisting in our body and they are providing different kind of functions. We will see that they are not only just existing, they are helping us to sustain life. They are helping us to perform day-to-day life activities, day-to-day life metabolism, and so many other things. So now some specific bullet points that is related to the human microbiome. So the human microbiome constitutes all the sides of the human body. As I said, not only just gut, not only just mouth, from top to bottom, it is everywhere. We will focus mainly on the gut, but the human microbiome is all over the body. So it is in the mouth, nasal cavities, throat, stomach, intestine, and urinary tract and skin. Everywhere. Microorganisms are everywhere in our body and also on our body. On the epidermis layer of our body also. So here comes a number which can be asked in your different examination like CSIR, DBT, and if you are giving any ICMR or any such exam. So the total number of microorganisms in the human microbiome I am telling is approximately 10 to the power 14. You can imagine how large number it is. 10 to the power 14 microorganisms constitutes the whole microbiome of a human body, which is about what? It is about 10 times more than the total number of human cells in a single person. Whatever cells we are having, the microbiome is about 10x time higher. So you can just imagine the role of the microbiome in protecting the human body. Now here comes the whole summary. We have to focus on this gut microbiome, but before that we also have an insights about what different kind of microorganisms are inhibiting the nasal cavity, the mouth, the skin, and different things. So you can see the most common one when anybody will ask you a question that which common bacteria is found on the epidermis of our skin. So it is none other than staphylococcus. So staphylococcus and its other species are widely found on our upper layer of the skin. So this is the normal microbiota of the conjunctiva, that is coagulase negative staphylococci, Haemophilus, and the Staphylococcus aureus. Staphylococcus aureus is the most common bacteria found on our skin. Apart from that, Streptococci. So this is about eye. In our eye, these four type of bacteria are commonly found. Now, apart from that, normal microbiota of the nose. What kind of bacteria we can see in our nose? We can see Staphylococcus, obviously, Haemophilus, Neisseria, Staphylococcus aureus, Streptococcus pneumoniae. We know Streptococcus pneumoniae causes the pneumonia. So all these things are found in our nose. Now, after the nose, mouth, and oropharynx. So this much of bacteria you can see: Streptococcus, Neisseria, Porphyromonas, Treponema, Fusobacterium, Diphtheroid, Haemophilus, and then again Staphylococcus aureus. So you should have an insight about what different diversity of microorganism we can observe in each and every organ. Like whenever a question comes that the eye is inhibited by which kind of bacteria, so you should know about that. Haemophilus will be there, Staphylococcus. If you don't remember the species, you can also write that Staphylococcus species, sp dot is enough. You don't need to remember so many species because there are so many species. Like in this you can see large intestine, so I can mark that we want to study about the normal microbiota of the large intestine. In the large intestine you can see Fusobacterium, Clostridium species, mostly the uh, food poisoning bacteria, Clostridium botulinum, which causes the food poisoning, and also E. coli, Klebsiella, gram-negative bacteria, Proteus, Lactobacillus, Enterococci, Actinomyces, Actinobacter. All these are large intestine bacteria. Now, this is an overview. We're gonna study it in a very detail about the exact category of the microorganism we observe in each and every organ. You can see here the normal microbiota of the stomach: Streptococcus, Staphylococcus, Lactobacillus, and also the Streptococcus. Apart from that, on a normal skin, on an epidermis, you can see again Staphylococci, diphtheroids like Propionibacterium acnejo, which causes acne, Bacillus species, Candida. Candida is a fungi. Candida albicans, which is a very common fungi responsible for so many kind of skin ailments and also the vaginal diseases. Now this is the normal microbiota of the urethra. So you can see so many different so many bacteria are common. They are not only just located to the mouth, they are not only just located to the large intestine or small intestine. In the vagina also you can see different kind of diversity of the bacteria. So this is an overall uh, summary that what different kind of microorganisms are present in our different organs. So now we will study in detail that the microbial community which are residing in an healthy human body do what? So these are the four questions we need to found the answer. Do individuals share a core human microbiome? If a person having a type of bacteria in his or her mouth, is it necessary that the B will also have the same kind of bacteria? Yes or no?
No, ma'am,
No. Why no?
because habit, environment, each and everything, fooding is different.
Yes, right. Absolutely right. it depends on the type of food habit. All it depends on the type of condition we are providing in our body. If we are consuming regularly acidic food, then acidophilic bacteria will be there. If we are consuming some sugary food, then there might be different bacteria or different microorganisms. So all it depends on the condition we are providing, because in the laboratory also we use different media. Like as a human being we consume food, so likewise bacteria or any microorganism, fungi also, they need different kind of nutrients to grow. So in the microbiology there are different kind of media which supports the growth of a particular microorganism. So if your body is rich in a particular kind of nutrient, then it will raise to that particular microorganism only, not every microorganism. So all it depends on the habitat we are providing in our body. So the microbial community in the healthy human was once considered to consist of microorganisms that were merely common cells, but we now know that this microbial community is important in early development and overall health and predisposition to disease. Now, why we are studying the human microbiome? Because nowadays, in the about a decade ago, it was only that microbiome is supporting life, but nowadays this microbiome is somehow altered, and then this is causing so many different kind of diseases. So why this is so, we need to have an insight of the total composition of the microbiome, that what does it consist of, what are the factor which is uh, affecting the overall composition of the microbiome, how it is converting in, how it is altered, how it is causing so many disease. So nowadays not only just composition is enough, but also we have to focus on what are the diseases that may result when the normal human microbiome is altered. So first question is this: do individuals share a core human microbiome? Yes, generalized structure will be same, but the abundance of microorganism will be different in each and every individual. Now, next question is there a correlation between microbial population structure and host genotype? Obviously, all it depend on the host. What kind of host? Even if some kind of microorganism, suppose a microorganism is in human being, it may or may not occur in any animal, let's say cow, goat, sheep, because again the same answer, all it depends on the environment we are providing. So there is a definite correlation between the microbial diversity and the host. Now, the third question: do differences in the human microbiome correlate with differences in human health? Obviously, yes, because if a person is always ill and having a bad intestine or a bad digestion, then definitely its normal microbiome is altered. So definitely the microbial population will be different as compared to the healthy individual, as compared to the healthy person. So recognition that these microorganisms function as mutualist means always positive, that play a central role in human health, has prompted formation of an international research program called Human Microbiome Project. Now, this is an important part. Human Microbiome Project is about recognizing different kind of microbial diversity. Now, there are certain terms which, if you will remember, you can explain each and every term to your examiner. Like, I have used the term microbial diversity. Microbial diversity means different kind of microorganism, A, B, C, D, and even in species A, B, C, D. So these are the microbial diversity. It means a human body are having different kind of microorganism in them. So Human Microbiome Project was a United States National Institute of Health Research initiative, NIH initiative, that is National Institutes of Health Research Initiative, to do what? To improve the understanding of the microbiota in the human health and disease section. Like how we can know the condition which are responsible for altering the normal normal microflora into diseased microflora. So definitely we need to know the factors which are triggering this transformation, this normal microflora to disease microflora. So basically NIH has focused to improve the insights of what is the composition of a normal healthy individual microbiome and a diseased person microbiome. So it is originally launched in 2007. It is originally launched in 2007. So the first phase was to focus and identify and characterize the human microbiota. Like I am saying that microbial diversity is there in the human microbiome. So obviously we need to know that what are the different kind of microorganism we can see in a human body. Suppose if in a class, suppose in class 12, we are having 60 students. So the class teacher should know about the major location that from where the most students belongs to, like whether the students are localized or whether they are from faraway places or from nearby states. So likewise we need to know that in the microbiome composition which is the most abundant bacteria and which is rarely in the healthy human microbiome, so that we can compare it with a diseased individual. So first phase was to identify the microorganism of a healthy individual, and then we can compare it with the diseased individual. So this was their first aim, to unravel the whole insights or whole composition of the normal microbiome of a healthy individual. So how is it possible? It is possible through the 16S rRNA gene sequencing. Now, many of us have you ever done 16S rRNA sequencing? Any of you?
No, ma'am. No, ma'am.
Okay. So I can explain this. Nowadays, you have to remember this 16S rRNA. 16S rRNA. This is known as the phylogenetic marker of prokaryote. 16S rRNA is present in the smaller ribosomal subunit of the prokaryote. So why it acts as a phylogenetic marker? Suppose you have bought some water sample from a river, pond, lake, or from anywhere. So from that water sample, you have isolated so many bacteria. Let's suppose B1, B2, B3, B4. Now, without identification, how will you able to know that B1 is what, B2 is what, B3 is what, B4 is what? So, in microbiology or in the basic research, 16S rRNA is the first step to do so. Before going on to the advanced research, we need to know that which bacteria is B1. At least we need to know about the genus, that this is from Bacillus, this is Pseudomonas, this is Staphylococcus, this is Enterococcus, and whatever. So for that reason, we do the 16S rRNA gene sequencing. Now, what exactly is 16S rRNA gene sequencing? Suppose this is a bacteria. Wait. From where I can use the dark screen. Okay. I will let you know because this is a very important part. Suppose this is a bacteria and this is its nucleoid. This is peritrichus. Peritrichus means all over the surface flagella is found. So this is a bacteria, And you all know the bacteria have a chromosome. Definitely it will have a chromosome. So you want to know the name of this bacteria. You want to identify the genus and if possible species. So how you will do that? So for this, a tool which is known as 16S rRNA. R is always smaller, ribosomal RNA sequencing, which is done through the most common one, Sanger sequencing. The most widely acceptable sequencing method is Sanger sequencing, which is also known as the dideoxy method. So what you will do to identify this bacteria? So in this whole chromosome of the bacteria, there must be a region which is known as the rRNA gene, which will be always unique for each and every organism. For bacteria 1, it will have some, let's suppose AUGCC. For bacteria 2, it is approximately 1500 base pairs. So for a representative purpose, I am only representing some few base pairs. Suppose AAUGCGC likewise. So this is the signature nucleotide sequence which is reserved, which is unique for each and every bacteria. Suppose you will have an ID card of your university or college or institute or whatever. So in that ID card, you will have your name, your parents' name, your roll number. So that roll number will be unique. Suppose your roll number is 211012. So this 211012 will be for only one student and not for other. Other will be 1314 and likewise. But the number will be different for each and every student. So likewise, this rRNA gene sequence is different for each and every bacteria. So that property we exploit to know the name of the bacteria. So what I will do, I just need this section. Which section? Out of the whole chromosome, out of the whole genome, I don't have to work with the whole genome. I don't need the whole genome. I only need this one, rRNA gene sequencing. I only have to work with this one. So what I will do, I will just amplify this region. So just quickly tell me, for amplification of a certain region, what technique is used?
On PCR.
Yes, right, PCR. For PCR, what do we need?
For like there are three steps: denaturation, annealing, and extension. For annealing, we need a
Yes,
bacteria.
bacteria. And what is the most critical requirement for amplification? Primers.
Yeah,
Primers. We need the forward and the reverse primer, right? So I have the template. The template is my this one. This region is the template, this rRNA gene sequence. So now, if you don't know this bacteria, how will you know about this section, this template sequence? So for that, in microbiology or in molecular biology, we have universal primers. Universal means it will bind to each and every bacteria. Necessarily, it will bind. So we use universal primer to amplify this 16S rRNA gene sequencing. Then after the sequencing, we have amplified this region. Out of this whole genome, we got the stretch of the 16S rRNA. Now what we will do? After sequencing, we will have some stretch of sequences, right? Suppose A, U, G, C, C, T, U, C, G, and likewise. Now, we need some repository to compare that whether this is reported in the literature or not. So for that, what do we need? We need to go for the BLAST. basic local alignment search tool. Basic local alignment search tool. This BLAST will do what? It will search this sequence as a template. It will search in its directory, in its repository, that whether this sequence is previously feeded or not. So if it will be feeded, suppose this is feeded as Pseudomonas aeruginosa. Aeruginosa. So it means that your B1 bacteria is Pseudomonas aeruginosa. It will show the similarity percentage. If it is more than 99%, then definitely your organism will be Pseudomonas aeruginosa. So I hope you have understand the whole mechanism, that how to uh, know the exact genus, and in many cases we can also know the species of the bacteria by using this simple 16s rRNA gene sequencing. Now this is a very basic step. This usually takes about in PCR 1.5 hours, like 1.5 to 2 hours is required. And after that we do the gel electrophoresis, let's suppose 45 minutes. And after that we purify the PCR, the gel DNA, and then we send it to the sequencing. So hardly it takes 3 to 4 hours to do the whole process. So we do this on a regular basis. So here comes the point. Human microbiome studies based on the survey of individual using 16s rRNA gene sequencing. So we use the 16s rRNA gene sequencing to know the genus or the species of the bacteria. So from the microbiome, we will extract the genomic DNA, that is the whole chromosome of the bacteria, and after that we will amplify the 16s rRNA gene, and then we will do the BLAST, that is we will use the bioinformatics, and then we can reveal the name of the bacteria. So through this method we can tell that this type of bacteria or this type of microorganism are located in this organ of an individual. So this is one technique, and nowadays this technique is automated. Nowadays so many automated sequencing machines are there, so many genetic DNA analyzers are there. And also apart from that, similarities between individuals are more evident at higher bacterial taxonomic levels such as phyla, and in the distribution of genes of similar function in the gut community. Suppose if you are isolating a gene from human being and from a bacteria, some common gene, so whether it will show more similarity or differences? Obviously it will show differences, because if you are extracting out from the same phylum or from the same group, then obviously it will show similarity, because we know the taxonomical hierarchy that if the phylum is same, more genetic similarity will be there. And if you will compare the gene A of human being 1 and human being 2, then there must be so many similarities, because obviously the group is same, the phylum is same, chordates are there. But if you compare one human being gene with a prokaryotic gene, then there must be huge difference. So the point is, we can expect more similarity if the group is same. Now, the possible outcomes of these analyses for clinical medicine include the development of biomarkers for predicting an individual predisposition to disease, the design of drug target, and the so-called personalized drug therapies. If you will reveal the whole microbial diversity of an healthy individual, then it will be very useful on a medical perspective, because in a healthy individual you can just say that in the gut A bacteria, B bacteria, C, D, E, F, G bacteria will be there. So if that healthy individual will start having some unhealthy food habits, then definitely this A to F will be altered. So this type of uh, technique is used to make personalized drug therapies. It means if a drug is suitable for me, if a drug has no side effect for my body, then it doesn't mean that it will be suitable for your body also. All it depends on the environment. So now we have to find out the answer of all these questions, like gut microbiota. In the gut microbiota, what kind of microorganisms are there? Which phylum dominates the whole microbiota? Which phylum is rarely found? And what do they do there? What is the function? And also, if they are influenced by some factors, we need to know about those factors in order to remain healthy. If I want to be healthy, if I want to stay alive for so many years, I need to know what are the some antagonist of for my body, what are the different factors which may alter my normal microflora. Suppose I know that carbonated beverages like uh, what Sprite, Coca Cola, and all those things are very much harmful for my body. So I need to know about that uh, through what ways it will alter my normal microflora of the body. So all those things which will be revealed in this module. Now here comes an important part. Before this, you need to know about this. So the human gut microbial community is that humans are monogastric and omnivorous. We eat both veg and non-veg. So humans are monogastric. You can see the human gut microbial community. We all have a different kind of gut as we compare the different vertebrates. So the variation on the vertebrate gut architecture. If the gut architecture is different, then obviously the microorganism will be different. All vertebrates have a small intestine but vary in other gut structure. So most absorption of dietary nutrients occur in the small intestine. This is a very basic thing we learn in the NCRT, that the major nutrient assimilation or absorption takes place in our small intestine. So in the small intestine, which microbial process takes place? The fermentation process takes place. So fermentation can occur in the forestomach, cecum, or large intestine. Now you can see here four gut fermenters. Example: ruminants, colobine monkey, marsupials, and hindgut fermenters. All it depends on the location of the fermentation. Microbial fermentation is taking place where? Is it in foregut? Is it in hindgut? So hindgut fermenters are cecal animals, primates, rodents, and some reptiles. So foregut fermentation is found in four major clades of mammals and one avian species. And hindgut fermentation either in the cecum or large intestine is common to many clades of mammals, including humans also. So point is, humans are monogastric. So what kind of microbial population we can observe in the humans? So a single chambered stomach is present in the humans. So in the human duodenum, ingested food passed down from the stomach is blended with bile secreted by bile is secreted by
Liver.
yes, bile is secreted by liver. So specifically hepatocyte secretes the bile juice, bicarbonate, and digestive enzymes. These three things are mixed with our ingested food, that is the bolus. And after 1 to 4 hours after ingesting that food reaches the gut, that is the large intestine, and by this time it is nearly neutral pH, and bacterial numbers have increased from about 10 to the power 4 to 10 to the power 8 through the microbial fermentation. Both the host and gut microorganisms share the easily digestible nutrients. The large intestine is the most heavily colonized area of the gastrointestinal tract. A point for the MCQ. Out of all the organs in our gut, the large intestine, the LI, is the most heavily populated by the microorganism, about 10 to the power 11 to 10 to the power 12 bacterial cells per gram. Now, colonization of an initially sterile gut begins immediately after the birth. Now, here comes a point. Before explaining this point, I will let you know that we have to also study about the infants microbiome, that just after the birth or just after the delivery, how it alters the, how they will develop the microbiome. Today only in the evening, I was uh, going through one post in LinkedIn. It was showing that the delivery method, delivery method, that is the vaginal and the C-section, also controls the composition of the microbiome. About this only I was going through it. It was very informative. I read that whole. I will share it with you. So they have. I don't know from where it was. The study was conducted. I think it was from USA or somewhere. So they have revealed that the delivery method, like if it is vaginal, the composition will be different. If it is C-section, the composition will be different. So you can just imagine that the birth delivery method also influences the whole composition of the microbiome or the microbial community. So in the microbiome section, the infant microbiome is a topic to be studied more and more, because it will be interesting that how an infant which is just born, just expelled out from the whole worm, so how it developed the microbiome. So we will study that also. So colonization of an initially sterile gut begins immediately after birth. A succession of microbial population replaces each other in turn until a stable adult microbial community is established. Obviously the infants have low immunity. That is why we need to take extra care of them, because they are under developing stage. They are not developed, they are under development. Their immunity is still under development. So the source of early colonizer is not clear, although some species are clearly transmitted from mother to infant. It is transmitted from the mother to the infants. As is now recognized for most microbial community, early description of diversity based on culture, microorganism greatly underestimated true diversity. Now, the most common gut bacterium is E. coli, the most common one. So, the entire phylum, that is the Gamma Proteobacteria, E. coli belongs to Gamma Proteobacteria, makes up less than 1% of all the gut bacteria. Now, E. coli will simply grow well in the laboratory. The most common gram-negative bacterium. It is a gram-negative bacterium and the most widely used microorganism for any basic research. So uh, they can be easily cultured in the laboratory and can thus be readily detected even when present in low number, because there is a specific test for the E. coli, the coliform test and all those things. So this is an structure of what kind of bacteria can present in the stomach, what kind of bacteria can be present in the large intestine. So the number of bacteria in the monogastric human gastrointestinal tract. The small intestine is composed of the duodenum, jejunum, and ileum. Numbers in the individual sections are estimates of bacteria per gram of the intestinal content in the healthy humans. So you can see in the stomach it is 10 to the power 4 per gram, and the pH is highly acidic because HCl is released, so it is highly acidic. Now it is duodenum, jejunum, and ileum, small intestine. So we have just now read that when we reach the large intestine, the pH become neutral. This point, neutral pH when we reach the large intestine. So we have also read that the large intestine is the heavily populated organ. This is heavily populated organs. You can see 10 to the power 11 to 10 to the power 12 per gram of the intestinal content. So here it is 10 to the power 4 and here it is 10 to the power 11 to 10 to the power 12 per gram, which is very much 10 to the power 4 is very much less than this one. So heavily populated is heavily populated is this one. So if an MCQ will come that out of the gut microbiome, which organ is heavily populated, then the answer will be large intestine. One MCQ is short from here, and the another one is 10 to the power 11 to 10 to the power 12. On the abundance, it will be asked, then it is the obviously large intestine. Now the second question will come that what is the pH in the large intestine, or what is the pH in the stomach? So slightly is heavily acidic is in stomach, and it becomes neutral when it comes to large intestine. The second question, so you have to remember. And the third question is monogastric. Human gastrointestinal tract anatomy question can also come. So humans are monogastric, single chambered stomach. So this section is important. So many questions can be asked from here. Now, the human microbiome is the collective genomes of microbes, whether it is bacteria, whether it is fungi, viruses, which are living in, and I have said in or on the body. So most dense in the gut. That is why we are studying about the gut microbiome, because it ultimately affecting our health. If the gut is healthy, we will be having a healthy life. If gut is unhealthy, we will not have an healthy life, because everything originates from our gut, like our immunity, our uh, what to say, central nervous system also act through our gut. We will see that how the brain gut axis is also a part of one of the module. So everything originates from our gut. So try to maintain a healthy gut habit, like what is harmful for our body, what not to eat, because we all are an educated individual now. We have an some sort of information, at least. It doesn't depend on the degree, like whether we are having UG or not, PG or not, or PhD or not, but we have so many basic information from everywhere on the internet. So we must utilize those information, because what's the point? We are studying but not incorporating it in our day to day life. We are consuming uh, whatever we want. We are not realizing that this is not good for our gut. So we should take care of all those things. So this is an overall what to say, composition of what kind of bacteria or what kind of microorganism you can observe in the gut. So firmicutes, proteobacteria, bacteroidetes, and actinobacteria. So what they do, what they are doing there, what is the specific function of proteobacteria, firmicutes, bacteroidetes, and this actinobacteria. So we will study each and every group in detail. So this is an overview, so I will not elaborate this here. This is an just slight overview that we will study this part. So now the factors impacting the gut microbiome. So nowadays, most of the factor will be at one side and lifestyle will be at one side. Our food habit, our sleeping habit, our physical activities level, like whether you are having a sedentary lifestyle or whether you are having appropriate amount of physical activities. So all it depends on the type of lifestyle we are having. Also the geography, at what location you are staying, and whether we are on some medication or not, because this pharmaceuticals or the regular medicine will have an 100% influence on the microbiome. Some bacteria will be inhibited by taking some kind of medicine. So pharmaceuticals, obviously the diet, what kind of diet we are taking, the level of stress, exercise, psychological stress, everything influences our microbiome. Don't just think that only your food habits, only your eating habits will influence your microbiome. No, it is not so. Nowadays, as per my opinion, stress is more dangerous than the any physical injury. If anything will happen to your body, you can still recover. But if more than one thing, or stress, anxiety, depression will have in your body, then you don't need to do anything. You just need to sit down and reduce your stress level, because your stress will impact your overall health up to a larger extent you can't even imagine. So emotional stress, mental stress, psychological stress, everything will influence the level of the microbiome we are having in our body. Now, just now we are talking about the infants. So infants, after the birth, they transmit the up to certain extent, they transmit the microbiome from their mothers, and after the birth process they slowly develop their own microbiome, slowly. So these are the major factors like diet, pharmaceutical, geography, lifestyle, and all those things will definitely impact your overall microbiome. So this is an important part, that we need to know about what are the different factors and how they influence that. Like acidic food will influence which part of the body most, like mouth or gut or anything. So we need to know about each and every factor in detail. So then only we will be able to know about the dysbiosis. So I will not complete this in today's, because we don't have time. So I will just provide an overview. The major phylum or the phyla of the human microbiome: Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria, and Verrucomo, by uh, what is this? Verrucomicrobia. So you will be able to see in Firmicutes what are the different kind of bacteria, in Bacteroidetes what are the different kind of bacteria. So also I have written the different factor which will influence the normal microbiome. So there are so many. So in a nutshell, what we have studied today: overview of gut microbiome composition. So the dominant phylum are Firmicutes, Bacteroidetes, Actinobacteria. We will study it in detail, don't worry. Variation factors are age, diet, geography and lifestyle, and also the pharmaceutical antibiotics if we are taking. So they have different role in our body. Gut brain axis, I have just now told you. Immune modulation, production of different kind of bioproducts, synthesis of vitamins, and digestion of complex polysaccharide. And we have to study this dysbiosis. Dysbiosis is a very important part, that how our normal microbiome just altered by different environmental factors. So that is known as dysbiosis, imbalance. So it will lead to inflammatory bowel disease, obesity and metabolic syndrome, neurodegenerative disease, because gut brain axis are uh, coordinates and act accordingly. So if gut is altered, our neurodegenerative diseases may also occur. Depression and anxiety. If our gut is not healthy, we'll not able to think properly. We'll not able to take decision properly. So everything is uh, coordinated. So this is all for the day. So you can answer me. Dysbiosis refers to what? Increase in gut microbial diversity, reduction in gut microbial metabolic activity, an imbalance in microbial community, symbiotic colonization of gut microbes. I just now I told you about this dysbiosis.
And imbalance in
Dysbiosis.
microbial
See,
community.
Yes,
Imbalance.
See.
right. See, correct. If you don't know about any term in biology, there is a trick. You need to break the terms. Biosis means what? Origination, living organisms, likewise. So this means alteration, failure. So dysbiosis means this normal biosis is somehow altered. So here is the meaning: an imbalance in the microbial communities. So you have to study each and every uh, multiple choice question from every topic to clear your concept. So I will take care of this. You don't have to do anything. You only have to join the session and you will have to listen it carefully, and we will together complete this module. So this is all for today. I need to stop the recording.